Let's spend a little bit of time talking about some basic structural features of viruses, the different shapes, different sizes, and how they infect in particular cells. And then we'll go ahead and we'll talk about a little bit about influenza. And so we know that viruses are small, right? In this case, particular poliovirus, we're looking at 30 nanometers, right? Which is much smaller, for example, than this E. coli, which is right here, right? Which is much smaller than a eukaryotic cell. So A, we know the viruses are the smallest, okay? There are thousands of known viruses, right? And so most of the ones we know about infect eukaryotes such as us. But viruses also infect prokaryotes. So bacteria get infected. Pretty much all types of organisms that we know are infected by viruses. Viruses play very important roles in the environment, which we'll talk about later, okay? So most diseases in the industrialized world that really cause problems are from viruses. And that's because we have a lot of vaccines, right? A lot of vaccines against bacteria as well as some viruses. And antibiotics work against bacteria. They do not work against viruses. And there's a lot less antivirals than there are antibacterial agents. And so there are other examples and other reasons why, but we'll leave that for another day. So viruses, remember, they are obligate parasites, which means that they cannot carry on their own metabolic pathway, which means that they need to infect a cell and then recruit that cell's metabolic pathways to increase their numbers. And we'll see that happen in the video that we watch. Okay, so they must get into a cell to reproduce. They all have genomes, and in this case, the DS is double-stranded, the SS is single-stranded, DNA or RNA, okay? And there may be several segments or single. For example, influenza has 8 single-stranded RNA strands. So know that, 8 single-stranded RNA strands, S-T-R-A-N-D-S. And this is just to give it a little bit of appreciation for the different shapes, the different sizes, right? Tobacco mosaic virus that infects tobacco is this long rod, okay? And that long rod is made up a whole lot of proteins. Right here, common cold, this is one example. There's over 400 viruses that can cause the common cold, okay? This is one example of it. And these are these little like isocahedral. Rabies virus is like a bullet. And then smallpox, which we've eradicated, huge success story for the world, right? Smallpox used to ravage lots and lots of people, okay? Smallpox, that's what it looks like. And all of these are under transmission electron microscopes, which is what the TEM stands for. And if you were to see an image like this by itself, just this image and said TEM and the size, you should be able to say whether it is most likely a virus or a bacteria or a eukaryote, because you'd look at that and you'd say, okay, 200 nanometers, well, that means that this one right here is maybe 400 nanometers, right? In which case, because you memorized the size classes of viruses, bacteria, and eukaryotes, you'd know, oh, that must be a virus. Okay, so that's a little clue right there. So in this case, what we're looking at is a naked virus up here where it's got a protein capsid and inside nucleic acid, and that is the viral genome. In this case over here, what we're looking at is a virus which infects bacteria. And so this again right here, this is the capsid, and inside this, so what's inside the capsid? That's right, whatever genetic material. And then right here, this base plate is what attaches to a cell, and basically the genome is transferred from the capsid through this tube and then into the bacterial cell to infect it with its genetic material. In this case over here, what we've got is a naked virus, meaning that the genome is inside a protein capsid, and that's the outermost. Whereas in this particular example, what we're looking at are enveloped viruses. An enveloped virus has a membrane on the outside, as we can see in these two pictures. Okay? So in this particular case, let's go into the middle first, where we know we've got a helical capsid, and we know inside the capsid is the genetic material. Exterior to that, what we've got is essentially these little yellow balls in here are individual proteins, and then outside of that, this orange with the blue ends that my finger's on right now, that is the envelope. That is the lipid bilayer, that envelope, okay? And so envelope viruses have lipid membranes. And one thing, the reason why that's important, this isn't just all academic, okay, naked, okay, enveloped, which of course you're sitting there thinking, well, this is really cool stuff. But functionally it's important because enveloped viruses can be killed typically with alcohol. So alcohol sanitizer, for example, can kill certain viruses because what it does is that alcohol can start dissolving some of that lipid away, thereby inactivating it. Whereas viruses that are just capsids and don't have an envelope around it, they're not inactivated by alcohol, okay? And so in this particular case, this is just showing two kinds where the capsid can be different. So the idea that I'm trying to show with these slides is not for you to memorize all of the different kinds, but no, genome surrounded by a protein capsid, and sometimes that's, and that capsid then have proteins on it, or outside of that there may be another membrane, and that's called an envelope. Okay, and what we see here are a couple of transmission electron micrographs of these particular viruses. And what we see in here is we can actually see that capsid inside that, which I think is quite cool. Okay, so in this case, we're showing okay, so it's a virus. How does it actually infect you? Okay, so we've already talked about the fact that viruses will adhere, right? And what do they use to adhere? Right? They use, I'm just going to use my fist here, they use a protein. So remember that whenever a virus is going to adhere and then infect a cell, it's got these proteins that are sticking on the outside of it, and the protein essentially is going to lock into something and allow it access into the cell. That is, if it infects. And so in this case with the naked virus right here, again, naked virus means it doesn't have what? That's right, it doesn't have the envelope. And so what we see here is that that's the case. So in this case, it binds to a receptor, and then it just sets in the viral genome. That's one example of a naked virus sending in its genome. Okay? In this case, what we're seeing is a virus that is what? It's enveloped. And we can see that because if we look at this particular image right here, what we see is that there's the genome surrounded by the capsid, and then outside of that is the envelope. Okay? So we see the envelope there. Okay, so what happens in this case is it bound with the receptors binding to the receptors, the ligands binding to the receptors, and then essentially it fuses with the membrane. So the envelope, know this, envelopes can fuse with membranes. Okay? And in this case, then what does it do? The payload is the capsid that's got the genome in it, and then the genome is released. And that's called uncoding. And we'll see a video, an animation of influenza doing this and uncoding and sending in its RNA. Okay? So then after it gets in, right, after it gets inside the cell and uncoats, well, let's not get into this. This is the process of endocytosis, but we'll let a video take care of that for us. Once it's in the cell and it's caused the cell to make a whole lot of other viruses, so viruses hijack the cell's machinery and they make the cell essentially, where they induce the cell to make more copies of the viruses. It's like a photocopier, okay, but it's really not. And so what this is showing is after the infection is ready, what we've got here is the viral capsid then goes out to the membrane and then essentially pushes against the membrane. And if you look at this, that membrane is coming from where? It's coming from the host. Okay, so if your cell is infected by a virus, for it to pop out, right, a new virus, it actually is going to coat itself with membrane that's from your cell. And so that's another way where it can kind of hide. Okay, and so this shows it budding off, and then this is showing the enveloped virion, and a virion is the term that is used for the entire envelope. So the capsid plus the envelope, everything is the virion. And most of us just shorthand that to say the virus. So let's take a moment to draw together a couple of very simplified renditions of the two general different kinds of viruses. And I mean general different kinds, I mean from a structural perspective. That is enveloped versus non-enveloped. So all viruses in the middle, they all have a genome, right? And so in this case, I'll just go ahead and draw the genome like this as one strand, and over here, I'll go ahead and draw it as multiple strands. Because some viruses have multiple strands and some have only one. Okay, now, what do we need to draw around each one of these genomes, right? What is it's made of protein, right, and it's held around it, and that is what we're looking at is the capsid, okay? And so in this case, I'm just going to draw the capsid like this for this one, and over here, we'll go ahead and draw it like this, okay? So now what we've got is the genome, and we've got a capsid, okay? So for the non-enveloped, that is the naked, we also need to make sure that we've got some protein spikes sticking out, right? Because remember, all viruses need to enter a cell in order to reproduce. They're the obligate parasites. And to do that, they need proteins on the outside, again, that will lock into a receptor on the cell so that the nucleic acid material, the genome, can get into the cell, okay? So that's what we've got for a naked or non-enveloped virus. And so if we go back over to the enveloped virus, what we're going to look at essentially is a membrane going around it, right? And what I'm going to do is I'm going to draw two. Maybe I shouldn't have used blue. Oh well. Hopefully you'll know that that's what we're looking at here is an envelope. And then what's sticking out of the envelope? What are some important components sticking out of the envelope? Look at this one over here. That's right. Some type of proteins. And there can be a lot more than just one type, right? But what we're looking at is there. And so, yes, are there some other differences between naked and enveloped viruses? Absolutely. But for the moment, this is all we need to concern ourselves because, again, we're just trying to get our heads wrapped around some very basic principles. So what we've got then in each case in green is the genome. And it can be single-stranded or double-stranded RNA or DNA. And there can be one or more of those, okay? And what we've got here is what? What is this? That's right. Those are the capsids, all right? What do we have out here? That's right, the envelope. And what is the envelope actually made of? What do we mean by that? Now I drew two to indicate the bilayer. So what type of bilayer is it? That's right, it's a lipid bilayer. And so lipid bilayer is more of a description on the chemical nature of it as well as the arrangement, a lipid bilayer. And then we've got essentially out here, we've got proteins, right? And these proteins, depending on what they are, they will bind to different receptors in the host cell. And so overall, what we're looking at is simplified, maybe make that a little bit lower here, simplified virus structures. And which is naked? This one, A or B? That's right. So this is naked. And this is enveloped. And so that's all we need to know about general virus structure at this point. So let's delve more into influenza. Okay, so influenza, historically, a huge killer. Okay, so I had a little bird, its name was Enza. I opened up the window and influenza. Okay, so this is a little ditty that kids would sing in 1918, 1919, when the United States, Europe, all over the globe was being devastated, right, by influenza. And at that point, they knew that it was somehow being transferred on the air. And that's where opening up the window, that's what that meant. Back then, they didn't know that birds were involved, like bird flu, because that could be another modern interpretation. Oh, they're talking about birds bringing in bird flu, but that's not the case, okay? So 1918, if we look at this, arrested for spitting on sidewalk Saturday. This is from a newspaper, and that's because they understood also that, secretions could transfer this. And so people were being quarantined, people were being isolated. And think about this, in the 1918 and 1919, like 40 million people died of influenza, right? About 675,000 Americans. And in terms of the global population, it's estimated that a third, 1 out of every 3 people in the entire world was infected by influenza. That is a massive worldwide pandemic, and it killed a lot of people, right? So what was going on in 1918 and 1919? That's right, World War I. So there were troops being mobilized and being transported all around the world for this war, right? Really sad time. Here in the United States, troops were getting put on these ships. People had influenza. They were sending them across the ocean, right? And on the ships, people were dying of influenza. Okay, so this, well, you'll watch a video. There's more information on the 1918 influenza that we're not going to talk about this because there's other media that will help you learn it. But we'll just leave it at that for now. So seasonal influenza, which impacts us coming up soon right now, right? And so seasonal influenza, signs and symptoms, we don't need to get into that. 1 to 4 days after the virus enters the body. In another lecture, I have 1 day, that's early. Some people it takes 4 days, okay? So treatment is supportive care. We know hydration, bed rest, right? Those are the key things, and if you can eat, do that. But basically, get in bed, drink a lot of water, and let it pass, okay? So Tamiflu and Relenza, they are antiviral drugs that you can take, but you need to take them within 1 to 2 days for it to have any effect, because there needs to be a low number. Once you've got a lot of influenza racking your body, that won't help. But if you take it early enough, then basically it cuts down the number of days that you are infected and have signs and symptoms by, on average, a day and a half. Okay, different people are different things. So the one thing I want to mention right now, so you know of Tamiflu and or Relenza. So the thing that you need to know now is they inhibit neuraminidase. Okay, so what is neuraminidase? We will talk about that more in depth, but that is a protein on the outside of influenza, and it is involved in allowing the influenza to escape a human cell. So after the influenza has been made inside a human cell and it needs to get out, right, it does so by using neuraminidase to cleave certain types of chemical bonds. Okay, so we don't need to go into the hardcore chemistry, but think of it as the scissors that allows the virus to get out. So if you inhibit the scissors, then it can't get out, and then it cannot infect other cells. Okay? And of course, we know that we've got a flu vaccine. And so the multivalent means that there's 3 or more strains, and that is strains, strains of influenza. Okay? So we know that you inhale it by the lungs or self-inoculation, and that's where people touching their face during flu season, that is a sure way to get influenza because you're touching doorknobs, you're grabbing stuff, you're out, you're at the ATM. If people have been touching that have influenza and they've contaminated it, right, then you're going to go ahead and get that. And that is an indirect contact, and what you touch would be the fomite or the fomite, right? So if you touch a doorknob and then touch yourself and you inoculate, that's how you can get it. Okay, that's where sanitizer being all around, alcohol sanitizer works really well. So why does alcohol sanitizer work well for influenza? That means influenza must have what surrounding the capsid? That's right, an envelope. Okay, so that's why it works for influenza, the hand sanitizer. Okay, so it is typically a lower respiratory infection in your lungs, but it can also be upper respiratory infection. Okay, it just depends. The temperatures between upper and lower are different, and different viruses are more suited to living in certain temperatures. Okay, so when a person is infected with, so say their lungs are infected with influenza, those cells are breaking down after they've made a lot of viruses. They're dying, which means that essentially you've got these little holes in your lining that shouldn't be there, which allows you to be infected by some type of bacteria or fungus or another virus, but bacteria or fungus, and that would be a secondary infection. Okay, so common secondary infection agents are Haemophilus influenzae, and the reason it's called that is because back in the day when they were using Koch's postulates and taking out bacteria and trying to find out what the causative agent is, they thought they got it. The scientists were like, yes, we've gotten this from all these patients that have the flu, right? However, it was from a secondary infection. So that's why it's got that name, right? And it is common, so that's another way where Koch's postulates actually break down because it's common, right? So if you normally have it in a healthy human and then your lungs start getting battered with influenza, well, it's there and it can go ahead and infect, causing a secondary infection. And then Staphylococcus aureus and Streptococcus pneumoniae are other common secondary bacterial infection agents, okay? And the thing about influenza as nurses, right, this can be quite debilitating, right? There's 200,000 hospitalizations every year approximately. And between 3,000 and 47,000 people die in the United States every year from just kind of run-of-the-mill seasonal infections, okay? A lot of the deaths are due to pneumonia and or influenza causes. So what you'll see often is P&I for pneumonia and influenza, okay? And other people die because they have underlying respiratory and circulatory causes. And so complications, can occur. And a thing to consider is that people that die from this, their immune systems are compromised. That's why they die from the flu. Either they're really young, right? So their immune system isn't fully developed, or they're on the older side, they're elderly, so their immune systems are compromised, or they've got certain types of immunodeficiency diseases. Anything that prevents you from mounting an efficient immune response can cause you to essentially die from pneumonia and or pneumonia complications, like secondary infections. So here is some slides from FluView. So cdc.gov, Flu Weekly, it's part of your reading, okay? This is an example of showing some of the metrics for, in this particular case, the 2016 to 2017 season ending in the 38th week, okay? So what we see then, the reason I'm bringing this up is we can see that it spikes, right? The numbers are really high in week 6. In week 6, that's in February, right? And so we know that the flu season is in at least North America, is typically fall through spring, and it hits really heavy in February. And that's from data such as this. Now this data is not the number of cases, it's not the number of deaths. What is it? Look at the y-axis. Anytime you're looking at data, look at the y-axis here. And so the y-axis in this case is the number of positive specimens. So what does that mean? Well, in this case, the CDC, the Centers for Disease Control and Prevention, this is what they do kind of every year, right? So they test more than 1 million patient specimens for the flu. We think it's the flu, let's test it, okay? Then about 90,000 specimens are tested in state and local public health labs. The CDC conducts full genetic sequencing on about 6,000 flu viruses every year, okay? So they test to see what type it is. Is it A? Is it B? Is it H3N1, H1N1? They do these types of genetic tests, and then they do the full genetic sequencing for about 6,000. Because we know that we need to be on top of influenza because any year they could mutate or it could do something called antigenic shift where all of a sudden you've got a pandemic type virus which is just going to clear house, infect a lot of people, kill a lot of people. And so by having this robust surveillance at the state, regional, and federal level, essentially scientists and healthcare professionals are keeping tabs on the genetic signatures of these different viruses so we know what we've got going on, we know what's being infected, and use that information for the next year's vaccination, right? So the viruses from this year, from this season, will be used to predict what may be growing next year, okay? So in this case, and that's down here, the CDC prepares, tens and tens of viruses so that when they decide which viruses they're going to use for vaccination, they're ready to roll. So if we go back to here, let's look at what some of these results are. So the red means what? That means it is type A. Okay, so now we're starting to get a little bit of the classification of the individual influenza viruses. And this is not just an academic exercise that's interesting in its own right, which it is, but what we're looking at is something that's very meaningful for understanding how this virus works and what we should look for to think about pandemics. Okay, so the predominant viruses this season is clearly H3N2. That is the most widely circulated from the specimens. And then if we go down here, then we've got, for example, the two greens are both B, Victoria and Yamagata lineage. The thing about the B classification, as opposed to the B do not cause pandemics. The A viruses are the only ones really that cause pandemics. So, And then what we have here is the number up here where if we look at this gold, right, that is H1N1. That's PDM09. That's the H1N9 from the pandemic, H1N1 from the 2009 pandemic, right? Hopefully we all remember that. That wasn't very long ago, right? So we see that that virus is still circulating around at low levels. So we've got mainly H3N2. We've got some of the pandemic strain, right, which a lot of people are now immune to, and it is in the vaccinations. And then up top is another green. So this is what we're looking at, just to know right now, in this particular season, 2016, 2017, what is the most common type of influenza virus in circulation? That's right, H3N2. And so this is just showing, the only thing I want to bring up that is, again, with vaccination, since we just talked about the CDC, right, and getting all the genetic signatures on this, what I want to look at now is just the fact that they select the strains for next year's vaccination in January and February. So in January and February, they're looking at this, they're looking at these types of data and saying, okay, what are the ones that we need to put in the next year's vaccination? And then they prepare it, everything, and then they're producing all the way until August, and then they're releasing and distribute. Okay, so just a little bit, so know what I just mentioned. Okay, so this particular one, now, first of all, anytime you look at data, what should you look at? So you look at the title, but then look at the axes. Okay, what is being measured? The measurement is always on the y-axis, right? In this case, percent of all deaths due to P and I. What does P and I stand for? That's right, pneumonia and influenza. Okay, so this is the percent of all deaths that are related to this. Okay, so if we look at these black lines, the lower one represents what? It's the seasonal baseline, and the real data is in red. So the number, the percent of deaths that actually happened in that particular week, right? And if we see down here the MMWR, that's for the morbidity and mortality weekly report, right? That's where these numbers come from, freely available. You're analyzing it in this class. We have the seasonal baseline, and we see that a lot of that goes right on prediction. So the seasonal baseline is predicted, okay, even though it's going up a little bit, that's not an epidemic level. At what point do we consider something at an epidemic level? Well, we've got this little buffer zone, and the buffer zone is actually determined by not just saying, let's have a buffer zone, but we won't get into that. So if it goes above this black line, that means that we've reached epidemic. Okay, so that's when it triggers. Okay, so if we look at that, are epidemics common every year? Well, look at 2012. Was there an epidemic? You bet. Look at the percentage of deaths due to P&I. That was more around what? Over 11%, and the epidemic threshold was like 8.5%. So that was clearly epidemic level, epidemic level, epidemic level. 2015, not a bad year, right? Still got to epidemic level, but not nearly as high a level of deaths, and not nearly for as long period of time, right? And then 2016, it's lower as well. And so every year, right, this is used to predict it. So be able to understand a graph that looks like this. If you're given this, you should be able to understand, you should be able to correctly point out what is being measured here, right, what is being represented, and that's the percentage of deaths. Be able to figure out a number that's pretty close, right? I mean, if it was around here and you said 9, that's fine, because right around here is the 9 range. So if we go 8, right, you can figure out what it is. And you should be able to know what week that is, okay? And by week, you should also be able to convert that to month, right? If it's week 12, well, we know that there's essentially, if we just break it up into 4 weeks a month, we know that we're, you know, January, February, March. We're near the end of March, okay? So we've talked about influenza. We talked about things like what Tamiflu and Relenza inhibit. What is it that Tamiflu and Relenza inhibit? That's right, neuraminidase. So if we look at this structure of a virus, it should look familiar because we've talked about structures of general viruses, okay? And so what we see here is we've got neuraminidase represented by this stick with these 4 balls on it, okay? So neuraminidase is on the outside. We know that it is like scissors where it allows it to cut a bond to get out of the cell, right? But how does it get in the cell in the first place? Remember, it's got a protein sticking out. So which of these do you think is binds to pulmonary epithelial cells? It's right up there. That's right, hemagglutinin. So hemagglutinin is another protein, and that's being represented here in the kind of, what is that, purple? Right? These are our hemagglutinin, right? And that's indicated right down here. That is the protein that will bind to the receptor on your cell, unlock it, and get into the cell. Okay, so hemagglutinin is the thing that allows it to get in. It's the so-called key. Okay, what we're seeing in here are strands of single-stranded RNA. So count up how many strands there are. How many are there? That's right, 8. Okay, and the reason I wanted to say that is you need to know that there's 8. And each one of this is showing what gene is on that particular RNA strand. Okay, so in this particular case, the hemagglutinin gene is on this particular RNA strand. Okay, the neuraminidase is on this particular RNA strand. These other aspects of the virus in terms of PB1, N40, NS1, don't worry about that. Very interesting. I cover that in my upper level microbiology classes, but for you guys, we don't need to go into all of the nitty-gritty detail. However, what we do need to know is that each one of these is written as a line. However, each RNA is actually wrapped up in other proteins, and that's called a ribonucleoprotein. Okay, and it's in your reading, RNP. Okay, so they're not just naked RNA, there's proteins actually wrapped around them, which are functional for some reasons. Okay, and of course, this blue is the lipid envelope, and as it's stated, and we talked about before, it's derived from the host cells. Remember, when the capsid pushes against the inside of the human cell, it pushes against the membrane, and when it's released, it's just coated itself in the membrane that your cell had on it, right? So it's leaving, you know, when an influenza virus leaves my cell, it's just coated itself in a nice blanket of Andy membrane, right? However, outside of the Andy membrane are the proteins. What are the two main proteins sticking on the outside? Hemagglutinin and neuraminidase. So this is just another picture from one of your readings. So the more, I think, the more different versions of these you see, the more you start seeing, okay, well, since there's only 2 types of proteins showing on the outside, I bet I know what that is. One of them must be hemagglutinin, which is showing up here, and one of them is neuraminidase. Okay? Let's not worry about the M2 ion channel, and then this is the ribonucleoprotein, which is represented in there. So let's take a look at this video, which is an animation of the influenza life cycle to kind of firm up some of the concepts we've been talking about. It's a great animation. So here we've got the virus. It is in, for example, your lungs. It is now binding to a lung cell. It's getting in. Your cell is now making a lot of viruses, and now they are coming out. And see, that destroys the cell, which allows for secondary infections. So the virion, again, represents the whole cell. And what we're looking at is 8 RNA strands wrapped up in proteins and then inside the capsid. Right? We know that always the RNA is inside a capsid. And that capsid is then, since it's influenza, it is wrapped in a membrane. And so what we're looking at, it's a type A for the pandemic strains. And it's moving because things move. And in this case, the person sneezed. And in each one of these droplets, look at all of those viruses. Okay, and that's what we're talking about here. So these tiny, tiny viruses, you can have lots and lots of them in these droplets. And this poor guy, he's just trying to read, right? He just breathed it in. And now what it's going to do is it's going to go ahead and bind to his cell and do what we just saw, and that is bind to specific receptors. That's what we see in the purple right there. Okay? And what is the yellow that's going to bind? What's the protein that binds? Hemagglutinin. So the hemagglutinin binds it. We have receptor-mediated endocytosis where it makes this package, and then it binds to this particular cytoskeletal filament, which is like a railroad, and it's going to be brought further into the cell. Okay, and that's what we're about to see right now. And so that's what we've got right now. This is a process that brings in nutrients, brings in other things that is hijacked by this virus. So now it is essentially the clathrin coat comes off, and it's about to uncoat because we need the genetic material. There we go. Boom. The capsid blows up. The 8 RNA strands are coming out, and they need to get into the nucleus. Okay? And so they're guided by processes that are not shown in this video, but what we see is that it goes in through these pores. It is now in the nucleus, and inside the nucleus are the enzymes to make copies of that RNA genome. So that's what's going on. In goes 1, and then 2 comes out. And then RNA is coming out, binding to ribosomes, and then proteins are being made. So now the ribosomes of the protein are making RNA, or are making proteins of the virus. Some of those proteins go back down into the nucleus, wrap up around the RNA, because remember, ribonucleoproteins, they then come out. More proteins that are going to be on the surface, like the hemagglutinin and the neuraminidase, are then leaving the nucleus. They're then being assembled. So this is the outside of the cell, and a virus is coming out. It's pinching off. Each one of these is pinching off from the membrane of the particular cell. And each one of those viruses can then go on to infect a new cell and start the process all over again. And what this is showing is antibodies binding to it. And another way your immune system comes, a white blood cell, it grabs the virus, pulls it in, and destroys it from within. So cool. When we talk about immunology, it just blows my mind. All of this does. Being here at the molecular level with all of these viruses is just fascinating. And so that is kind of a nice way to look at essentially the virus in action. So make sure you look at that link. When you click on that link through Blackboard, then there you have the regular person's narration. So hopefully between watching me talk about it after we just talked about this material and somebody else narrating it, it will really, really sink in. Now that we've seen that cool animation, let's talk about some repercussions. Because not only is what we saw for the life cycle, how the virus gets in and then makes your cell make a whole bunch more viruses to get out, but it has very important implications. So if we look at this particular image on the right-hand side, what we see is this is an animal cell, a human cell, and we've got 2 different strains of virus, maybe H3N1, maybe H1N1. So if that's H3N1, and then this one over here is H1N1, both of them get in, just like we saw. Both of them uncoat, right? Release the RNA in there. All of the RNA from both of the virus goes into the nucleus, right, which is what it's showing in here. And then when the RNA is made, right, more copies are made to make more viruses, now you've got 2 sets of the 8 strands of RNA, right? One from virus A, one from virus B. And what happens is when they're packaged, they're not packaged so that all of the RNA from one virus is packaged into the same membrane or capsid, and the one from the second type of virus is packaged into the other. No, it's random. Each virus that is essentially assembled inside the cell has one of the first RNA molecules, one of the second, one of the third, one of the 4. So basically what happens is you get a mix and match. You have some of the RNA strands that are from virus A and some of the RNA strands that are from virus B, and that is called antigenic shift. And that can create a new or modified virus, and that often is what causes a pandemic. So remember, in terms of influenza A types or B types, A is the one that causes pandemics, and it's the one that undergoes antigenic shift only. So that's the one that we really need to worry about, okay? What we're seeing in the left-hand side is mutations. So antigenic drift is when mutations in individual nucleotides occur, when all proteins, so if we look at, say, hemagglutinin, okay, if this is the hemagglutinin protein, that was encoded for by a gene. If that gene gets mutated so that one of the amino acid changes, well then maybe it looks like this, right? Maybe there's a new amino acid, so now we've got this thing that doesn't look like the original hemagglutinin, so it is mutated. It has a different feature that may make it not able to infect a cell, it may make no difference, or it might make it very much able to infect a cell or a completely different kind of cell. So mutations occur all the time in types A and B, okay? So that's just a part of the process. Mutations are always occurring. Whenever viruses are made, mutations occur because there are mistakes that are made, okay? That has a ramification that sometimes the proteins can change. Sometimes those proteins can mean increased infectivity or decreased infectivity or make no change at all, okay? So that's the difference between antigenic drift and antigenic shift. Which is the one that makes a more dramatic change immediately? That's right, the shift. Okay, so don't worry about these images down below with these bar graphs. Okay, and what's important is that this shift can occur in the same host, and it can come from different animals. So an organism can get infected by a human virus, can get infected by a pig virus, and they can assemble and mix to create this crazy Frankenstein, hybrid virus. And that's just the way it is. So let's go ahead and take a look at that more closely using the light board. All right, let's go ahead and work on something together. And what we're going to do is we're going to talk about antigenic shift of influenza. So let's talk about the genetic material of influenza. So influenza has what as the genetic material? That's right, RNA. How many strands? 8. You got it. So 1, 2, 3, 4, 5, 6, 7, 8 strands of RNA. Surrounding the RNA is the capsid, and surrounding the capsid is the lipid bilayer, right, which I am drawing kind of a little thicker and a couple layers so that you can see that. And then sticking out of this, right, what am I drawing here? And I'm just going to draw a few because I don't want to spend too much time drawing a whole lot. Okay, so what I'm trying to draw here is that these circles are the HA and the triangles are the NA. And so when we're talking about that, what we're looking at is essentially HA is hemagglutinin, NA is neuraminidase, right? And in this particular case, I'm just going to make up a hypothetical example. This is influenza of type H1N1. And let's go ahead and make that green to match. This is H1N1 over here. Okay? So H1N1 means that this particular virus has the first variant of hemagglutinin and the first variant of neuraminidase, which means that there's more than that. As a matter of fact, for hemagglutinin, there are 16 variants. Neuraminidase, we know of 9 variants, which means that there's H1, H2, H3, H4, H5, H6, all the way up to H16, and neuraminidase 1 to N. So that's how you can get the H1N1, H3N2, H5N7, whatever the case may be, it is that particular match. Okay, so here we've got H1N1. Okay, let's go ahead and draw another variant. Let's go ahead and draw, let's say, well, let's just draw it first. This is a different variant, so I'm going to use a different color. So it's built the same, so we just need to draw the same exact thing, except this yellow marker looks green for some reason, so let's get rid of that and use this nice pink. Okay, so 8 strands of RNA, 2, 3, 4, 5, 6, 7, 8. Okay. Then we've got what surrounding it? That's right, the capsid. And then what around that? The envelope, the lipid bilayer, right? And now again, there's power in repetition. The more we rehearse and we say out loud what it is we're drawing, the more you're drawing it and labeling it, the more you know it, and you don't have to study it more just by reading about it, right? So ideally, doing this along with me and following along, writing down notes goes a long way towards that. And so let's go ahead and draw the hemagglutinin and neuraminidase, right? And so I'm just drawing these however they are. There's no significance about having this one over here and this one over there, whatever the case may be. Okay, so again, what we're looking at is, in this case, the neuraminidase and this as the hemagglutinin. Okay, so we've got 2 different cases, H1N1 and over here we'll just say H3N2. Okay, so antigenic shift is when 2 or more viruses infect the same cell and out pops a mixture of both of them, as well as out pop, some of them can be the same exact ones. So remember, when we're talking about a cell, and what I'm drawing right now is the barrier for a particular cell, okay? So if each one of these viruses goes into the cell, we know that ultimately, right, If you think about the video that you watched, where the virus binds to the receptor, right? The lipid fuses, sends in the capsid. It then goes down that cytoskeletal filament, and at some point it uncoats, and what we have essentially is the RNA molecules in the cell. Okay, now when we say 8, they are 8 different types, right? So this is the first one, second one, third one, etc. So when a new virus is packaged together, it needs to have one of the first, one of the second, one of the third, one of the fourth, one of the fifth, one of the sixth, one of the seventh, one of the eighth. Okay? And when it gets assembled, there's no mechanism to say we need to package all of these together because they're one virus and all of these together because they're another virus. No, it's physics and it's chemistry and some biology that packages it up, and it just needs, I need one of these, 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 etc. Which means that when a new virus is assembled, right, and put into its capsid, we could get examples of that look something like this. Okay, so in that particular instance, what we could have is we could have a virus then that is made ultimately, well, that is made okay, let's stop for a moment. We're gonna continue with this, or just take the time code. Okay, I'm ready whenever you are.
Okay, ready.
So when these are packaged, basically, you just need to package so that you've got one of each kind. And in this instance, I'll just go ahead and say that one is packaged so that it's got the first one from the H1N1, the second two from H3N2. So that now is a new complement of RNA molecules. Another one that could be made is, for example, something like that. Okay? So what we're seeing is you can get a mixing and a matching. And now when these are assembled inside, right, they will then get a capsid inside the cell, as we saw in the video. Okay, but now the lipid bilayer, where is that going to come from? So what we've done is we've just assembled two, because these don't stick together, they mix and match. So let's go ahead and show the cell here. Okay, so if this is the human cell, right? So for these to come out, right, what's it going to pick up on the way? Right, what are we going to have come out? This is going to push out, and it's going to get the lipid bilayer from your cell out. It's covered in the video, so I'm not going to go over it again, but I just wanted to remind you that ultimately, when these come out, so when that comes out, it's going to look something like this. So let's draw this virus particle after it comes through. So let's go ahead and draw the proper RNA content. All right, now what color should I draw the lipid envelope? That's right, it's coming from the human cell, so that's what we're drawing right here. Okay, so now we just drew the same one here. So what are we getting at here? Well, let's consider that essentially what we have is a, if this first RNA strand encodes for the hemagglutinin and the second RNA strand encodes for the neuraminidase in both of these, right? Then when we get this particular one over here, right, what is this? What color are we going to draw the hemagglutinin and the neuraminidase out here? So which hemagglutinin is this? Did this come from H1N1 or H3N2? It's green. It came from this, so we know it's H1N1. So which means that let's go ahead and, for the hemagglutinin, we're going to go ahead and draw the green hemagglutinin, because it came from the H1N1. So we know that this strain is going to be H1 something. Okay? So now if we look at the next one is NA, and that came from H3N2, which means that this is an H1N2 variant, which means that we need to draw the neuraminidase from that particular strain. Okay, so when we have this is now an H1N2. Was there an H1N2 that infected the cells to begin with? No, right? And that's the power, and I think the beauty, although, you know, it's bad news, of this assortment, this antigenic shift. Okay, so what about this one? Let's go ahead and finish this out in terms of what this would represent. So if this one came out, right, so what we have is this one popping out there, and this one popping out here. And what we've got is essentially, let's draw this complement now. 1, 2, 3, 4, 5, 6, 7, 8. Let's give it its capsid, and let's give it its membrane that came from the human cell. And now let's go ahead and figure out what the H and N is. So the first strand of RNA came from H1N1, so that is H1. So let's go ahead and draw the H1 spikes. And now the neuraminidase is on the second strand, and that right there also is from H1N1, right? So it's green. So this comes out, it's still H1N1, right, which is one of the ones that came in, but is this H1N1 exactly the same as this H1N1? No, and that's where we can get a whole lot of variation. All H1N1s are not the same. All H3N2 are not the same, because when we refer to that, we're referring to only 2 of the RNA strands of the 8, because there are other proteins that are encoded for in these other particular RNA molecules, and so the variation is more than just the H whatever and whatever. Okay, so this is antigenic shift. Okay, antigenic shift. And so when we consider antigenic shift, think of shifting into a different gear, right? And that's what we have over here. And this one should say, excuse me, H1N1. Okay, so antigenic drift is something that's occurring really throughout this whole process. And antigenic drift is basically mutations that occur when these are being copied in the cell. So for example, an antigenic drift could amount to essentially one nucleotide change, right? From there to there, maybe there's just one particular portion that's been changed. And so now that's a mutation. Okay, so anytime that goes on, that wasn't even in any of the original ones, right? Whereas if you look at what I drew over here, at least each one of these RNA strands was exactly the same as what went in. It's just that they got mixed up, they got shifted and assorted together. Here what I'm saying is that a mutation has occurred. So there's like a one point where instead of one particular nucleic acid, it's been changed, right? And so, for example, instead of an A, maybe it's now a G. Okay, and we'll talk about genetics later on, okay? So if this particular RNA strand got put in, so this one right here, maybe this one came from the original one, or maybe it actually picked up there. And so what we're talking about here is this third one has this particular mutation, and that then goes out to here, right? And so now what we're looking at is an H1N1 that looks quite different from this in the fact that, well, it's got a whole different RNA strand from H3N2. So what it's encoding for is the same protein, but it's a slightly different flavor, maybe a different shape, may function a little bit differently. And there's a mutation in this other particular gene. And so mutations occur on a regular basis when viruses are reproducing. Some mutations cause no issues at all, no changes to the functional proteins. Others can cause big changes in the particular function of it. Okay, so antigenic drift is based on mutations. Antigenic shift is based on reassorting different RNA strands from different particular viruses that infect the same cell. In this particular case, well, maybe not in this particular case, but antigenic shift, we believe, is involved in all of the pandemics that have occurred. But anytime you have shift, you also have drift, because drift is occurring all the time as well. So make sure you get this general concept down. There's a really great video posted on the website that's more of a cartoon, but I think the power inherently of you doing this with me, writing it down as we do it, and then watching those videos, or vice versa, whatever order, is the way to strengthen this material so that you have the core concepts in your mind in that shift. So now that we understand how we can mix and match different viruses, and we look at this particular screen, it all becomes very clear. And this is the evidence that we have from H1N1, 2009, how it ended up being the virus that caused that pandemic, right? So how many different viruses was it made from? Look at this. The evidence is that there was a pig that had already antigenic shifted and already made an assortment of this new virus that came from classical swine virus, North American avian virus, and human H3N2 virus. Think about that. 3 different types of influenza viruses: classical swine, North American avian, and human H3N2. That had already been assorted and was in this pig. And the evidence shows that this Eurasian avian-like swine virus then infected it and changed it even more, and then that was transmitted in Mexico to a person, and that's what started the 2009 pandemic. And the reason we know that is because scientists have sequenced the genes. Remember, the CDC sequences full genomes in a regular year for 6,000 of the specimens, right? I don't know what the number is for 2009, but I'm guessing that they may have fully sequenced even more than that. Plus, it was being sequenced throughout the world because it was a pandemic. And so the forensic sleuthing that occurs is due to modern molecular biology and genetic techniques. And so, So we'll just kind of leave off on this notion of we're not going to talk about bird flu yet. We'll talk about that later. I just wanted to get the fundamentals of viruses and then a particular study, the flu, right, on what that looks like. But I do want you to know this, and that is how, for example, backyard poultry could spread bird flu to people, okay? And we say backyard poultry at the CDC because this is for the United States, right? We know that there are plenty of societies throughout the world where chickens are just running around, right? And so that is even more common for people to get infected because they're just all around the place. And so, but backyard chickens are becoming popular. Here in Jacksonville, backyard chickens were starting to be allowed, I think, a couple years ago. And so basically, if this bird is infected with bird flu, well, basically, it is coming out when it flaps its wings, when it scratches, shaking around, it's in its feces, and so handling it because you love your chicken or just breathing it in can cause you to get this particular type of influenza. And so nowadays, there's a big push for One Health, and that means that a lot of these diseases in the United States, well, a lot of these diseases that we're seeing, infectious diseases, shouldn't just be doctors, nurses, and scientists that deal with human infection diseases, but we need to have the veterinary doctors, right, involved. And so by having veterinary medicine also investigate these from the animal side and the human side, we're coming up with a lot more information that otherwise we would not have gotten, because what we're learning more and more and more is a lot of these diseases that are causing pandemics we're getting from a jump from an animal to a human. Like SARS was a jump from bats to civet cats to humans, and then human to human to human, right? This one right here, the 2009, came from a pig to a human, right? So more and more important, and that ties in with the section on emerging infectious diseases.